What I would like to share with you today is a topic that we work on in neuroscience on the brain. And in neuroscience, we usually see the neurons that are the nerve cells, but there are other cell types. So the cells I'm going to talk about and the process I'm going to talk about is called myelin. And myelin is important, as you will see, for the function of our nerve system. To present about myelin, and so first I will tell you what is it, what is myelin, what it is important for. Is it important for how it is working in our brain, how it is involved in different neurological diseases. And we know today that it is important for also aging, for longevity, and for different degenerative diseases that are associated with aging. And of course, the last thing is whether if there is anything else that we can do in order to improve myelin and brain health during our aging process. Maybe I will start by an example. Why do we need myelin? We need myelin in order for our nerve system to work very fast. And just as an example, I think you all know this guy who, I don't know if you know, but he's pitching in a speed record. So he throw the balls at more than 160 kilometers per hour. Okay? This is almost the world record, by the way. And which means that it has 0.4 seconds or 400 milliseconds for the ball to reach the batter box. Okay? So this is 400 milliseconds that this guy that hits needs to calculate how the ball is coming to it, to coordinate the movement, and then to activate the muscle and hit the ball. This is only 0.4 second. And this is really one example. In fact, the need for speed in life is actually more important in wild, right? Because there are animals that need to have a very fast flight response or escape response. So that's why you really need the speed during evolution. So how do you make fast conduction or fast signals in our nerve system? And let me just tell you what is the problem. The problem is that, so this is our nerve system. It is divided into two main parts. The first is the central nerve system that contains the brain and the spinal cord, and there are all the other nerves that are coming out that are called the peripheral nerve system. What is the issue? The issue is that, for example, a nerve cell that is called a neuron that is present here near our backs send processes that goes up to the central nerve system, but it also sends processes that goes all the way to our feet. And if you draw such a nerve cell, it looks like this. This will be the cell body here, and it will send huge, very, very, very long process, and the information needs to get from one side to the other very fast. Okay, so this is, for example, a neuron that sends heat, for example. If you have some heat here, it needs to tell the brain that it is hot. So there are several ways where you can make things very fast. So one thing is you can make the neuron. So a neuron is the nerve cell, right? It has a cell body and it sends a very long process. This process is called axon. So one possibility in nature is to make these axons bigger. So there are some animals that do that. So the squid, for example. The squid has a neuron here in the brain and an axon that goes here back. And this axon is just huge, okay? And it allows this to swim. So this axon is extremely big. If you look at, will be how fast these neurons will transmit their signal. Big axon, which is like 0.5 millimeter here, will transmit at 20 meter per second. So do you think 20 meter per second is fast? Not so much. And the other solution is, instead of making the axons bigger, you just take them and you cover them with insulating material. And this is exactly myelin. So myelin is a material that will insulate, will wrap around these axons and will allow fast conduction. So now we can compare a nerve without myelin and with myelin. Okay, so before we saw this is 600 microns. This is 0.6 millimeter axon, huge axon. It will conduct at 20 meter per second. Now, if you go to even just 8 micron myelinated nerve, it will conduct at 80. So it will conduct 4 times faster. And before you translate, myelinated nerves go up to 200 and 240 meters per second. So now, if you think about it, what is the main advantage? It allows, now you can use much thinner axons, right? Instead of much bigger axons. And this allows a lot of nerve cells to be placed in a box, our head. So it allows a lot of neurons to be placed in the human brain. And when I say a lot, because the human brain contains about 86 billion of neurons, plus another 86 billion of different cell types. Because otherwise, this is what we'll have. So otherwise, we'll have to use a huge head. So this is a very important point in evolution that allows making a brain. And to show the complexity of the brain, I'm going to show you a very short movie that there is a method where you can take a tiny piece of the brain and cut it and then look at it with electron microscope, okay? And then you can take a section and then cut another section and another section, and like this you can come and make a 3D reconstitution of the tissue. So this is just 2 microns, but look at how many cells there are there. But just to show you how complex our brain is and how crowded it is. So let's go back to myelin. What is myelin? So what we have here is we have a neuron. This is a nerve cell. It has here the cell body. The signal is coming from here, and it goes along the axon until the target. So myelin will be this ensheathing material, insulating material, and if you make a section here through the myelin, so you make a section, and then I turn it like this. Turn it, and this is what you see. So you would see here, this would be the nerve cells, or the axon, and this would be the myelin. So the myelin is not one wrap, but it's in fact a lot of wraps of a membrane of another cell, that we'll come to that in a minute. And if you look at electron microscopy, this is real nerves. This would be the axon, and this would be the myelin. It is produced by, so this is the peripheral nerve system, and this would be the central nerve system. And myelin be made by two different cells, Schwann cells in the peripheral, and oligodendrocyte in the central nerve system. Since we are going to talk mainly about the central nerve system and the brain, this is how it looks. This is a picture of neurons stained in red here, and this is in green. This would be the oligodendrocyte, this cell that makes the myelin, and you can see it send processes and make different myelin segments. Myelin is being composed of a lot of lipids. There is a lot of lipids. There is maybe 70% of the structure is a lipid, and lipid looks white. That's why we all heard about that in the brain we have so-called gray matter and white matter. Okay, so white matter is being is basically parts of myelin, long stretches of myelin, and it looks like this. So also in the gray matter there is myelin, but it looks a little bit different. So myelin, in fact, allows very fast conduction, but it also allows very fast working of nerve cells. I'll try to explain why do you need myelin here. Okay, so what myelin allows. So what I'm showing you here is a neuron, an axon here, that has several parts of myelin. Between the myelin, there is a region that is called the nodes of Ranvier. It's on the name of a French anatomist that discovered these structures. And in these areas here, there are certain types of engines that allow the nerves to signal. And these engines are called certain channels. They are called sodium channels. So there is a lot of channels here at the nodes of Ranvier. And the signal, the nerve signal, since it cannot go here because this is insulated, it will basically jump from here to here to here. And this is called saltatory conduction. You can look at this structure that is very similar to transoceanic cable, transatlantic cable. It's the same principle. If you have a cable with terminals here, it will go under the water. It needs to conduct the signal. It needs to be isolated. And it also has these repeaters that generate the signals. Okay? Very similar to this structure here. This is the first thing myelin is doing. It is making the nerves work faster. It is also saving a lot of energy. And you want to save a lot of energy because, I don't know if you know, but our brain requires about 20 to 30% of all the energy that our body expend. So it is also reduced the energy for conduction. It also saved the space. We talked about it already. And the last function of myelin is to give support to the nerve cells, give support to the axon. It gives trophic support, help it to be alive, but it also gives a metabolic support. So this system, so let's talk about this a little bit more on metabolic support. It means that this system, this myelin and nerve cells, is very romantic system, extremely romantic. There is a dependency between the nerve cells, between the axons, and the oligodendrocytes. And why this is important? It is important because if you have neurons nerve cells that had myelin, and now you take the myelin away from them in disease, for example, or injury, eventually these axons and these neurons will die. So myelin is not just static structure. There are different ways that myelin can be regulated. So nerve cells can be either non-myelinated, they can be partially myelinated, fully myelinated, or their myelin can become in different types and shapes. And these changes between this and this, for example, or between the thickness of the myelin can be regulated by the brain activity. If we have so this is experiments mainly that came with experimental animals like mice, for example. You can activate certain neurons, or in different paradigms of learning and memory, or both sensory and motor. You look now at myelin. So what you see, what people saw, is that during the learning process, myelin can be changed. You can have new myelin being made, changing of other myelin fragments that coming out and coming in, and it's a very so-called plastic event. So activity and brain function can change myelin. Can myelin itself change brain activity? So sorry, before I answer this question, this is an example of how our activity can affect myelin. So people have seen that in people that are piano players, and especially piano players that practice very, very early in their life, there is different changes and organization of the myelin. In fact, they have more myelin. So now we are coming to the question whether if you change myelin, can you change behavior, learning, cognition, and the brain activity? So people or scientists have used different paradigms. So you can teach, for example, animals to do some task and then to change myelin and see whether they are good or bad at it. And I will give you one example. And this example, I love this example. So if you take mice and you let them run on a wheel, they will run, right? They love to run on a wheel. But now if you take the wheel or the ladder, right, and you take out some of these and you change the wheel itself, now it takes them time to learn how to do it. So what certain scientists in England did was to now, by some genetic means, not allow new myelin to form, okay? And he asked whether if you stop myelin to form, whether you can learn this motor skill. And the answer is no. So if you block, and this is not newborn, or this is old animals. In old animals, you still need new myelin to form in order to learn motor skills. This means that even a small change in myelin throughout life can have a very important function on cognition and behavior. So I convinced everyone that myelin is important? Good. So it's also important, of course, for different neurological diseases. If there is a problem in myelin, we get problems in our functioning of the brain. And I will give you some examples. If you have big issue with myelin, let's say you have a genetic defect that have no myelin, or you have a disease where the immune system attack the myelin, like multiple sclerosis. This is easy to understand, right? No myelin disease. slow conduction, disease. So these are all called white matter diseases, and it contains different type of, it's called leukodystrophies, multiple sclerosis. In the peripheral nerve system, it's called neuropathies. But in addition to this, so these are very tough diseases that have no myelin, or myelin is disturbed over there. But we know today that abnormalities and dysfunction of myelin is present in almost any disease that we know. So we know that there is a lot of involvement of myelin in diseases, and there are a lot of diseases that are associated with age. So let me go through a little bit what happened to myelin with age. So myelin starts in a way after we are born, or in the third trimester when we are embryos. So it starts postnatally, and then we get more and more myelin in our childhood here, and when we grow up till the age of 20, about the age of 20, and myelination continues to our 40s. But then, after 60, 65, this is my age now, at around after 60, you start to see deterioration of myelin. And in fact, I know that because we work on mice, and if you look at the brains of like old, very old mice, you always see it's kind of normal. You see abnormalities in myelin. And this reduction in myelin is also associated and linked to memory impairment, and is correlated with the age-related cognitive declines. So this is so no wonder that there are many age-related neurological diseases and neurodegenerative diseases that are linked to myelin, and there is a myelin involvement. So I'm not going to read all this, but just to say that, myelin is now known to be involved in Alzheimer's disease, in Parkinson's disease, in frontomental dementia, and other diseases that are associated with age. So what can we do? And let's just take multiple sclerosis as an example, right? There is loss of myelin, and now what can you do? You want to prevent axonal degeneration, and you can do this by so-called remyelination. So the idea is to somehow enhance myelin and to protect these axons from dying. So I'm not going to go through the details here, but there are different strategies that you can interfere at different points and help make new myelin. Yeah, you know what I will say? For example, you can make the cells that make myelin, you can try to make more of them, or you can somehow allow these cells to, by different drugs, to make more myelin. And this is something that a lot of drug companies are trying to make. The question, really, do you need drugs, or there are other things that can help you save your myelin and induce your myelin? There are two things. So there is the effect of diet, there is effect of fasting, there is a lot of effects. Two things I want to mention that I think are important, and one is physical activity, and the other one, meditation. So physical activity, for example, and also in old age, promotes remyelination, especially after injury, and it's also preserving the white matter, the myelin. It's preserving our myelin and stopping it from deteriorating. And really the same thing goes for meditation. So there are a bunch of papers over there that people have tested this. And again, you tested it with MRI and also experimentally. And already after several weeks of meditation, you see changes in white matter that indicates that there is something positive that is going on. meditation and mindfulness, for example, and yoga all have another effect because they affect our frequency of the brain working and the activity, so-called the rhythmical neuronal activity. And this rhythmical neuronal activity also have a positive role on the oligodendrocytes. And since this is a very romantic system, the neurons are affecting the oligodendrocytes are affecting the neurons, and like this you can save the brain integrity. And I will just finish with the slide that now you can know what I choose. So what I do, sport, aerobic exercise, and yoga, which also include meditation. And what you see here, in fact, is imaging of the brain. This is the white matter track of the... And I will stop here, and I will be happy to discuss and answer any question.